In this hour, we are highlighting bladder cancer. Two new members and a senior member of our organization will present on bladder cancer. First one is Martha Terris, a new member, to talk about newly discovered HYAL4. V1 chase drives gemcitabine resistance and predicts chemotherapy failure in bladder cancer patients. She is followed by Cheryl Lee, another new member, on genomic dynamics of immune and epithelial compartments during murine bladder carcinogenesis, implications for sex bias. Finally, Dan Theodorescu, he's going to talk about enhancing cisplatin therapy in bladder cancer.
Hello, I'm honored to be here today to talk to you about our research into chemotherapy resistance in bladder cancer patients. This research is spearheaded by Vanetta Lokeshwar, the PhD that is chair of our biochemistry and cell biology departments here at Augusta University and is an outstanding scientist. First, for the disclosures, our funding is from the NCI and the Department of Defense. Everyone here knows glycosaminoglycans. They're synthesized by the urothelial cells, and the glycosaminoglycan layer or GAG layer in the bladder is protected. Defects in the GAG layer have been implicated in interstitial cystitis, and treatments for this disease have been aimed toward replenishing this layer. The layer is composed of unbranched polysaccharides, repeating disaccharide units such as the unsulfated hyaluronic acid, and proteoglycans. The GAG layer is degraded by an enzyme family known as hyaluronidases. Hyal1, 2, 3, and pH20 degrade hyaluronic acid. Hyal4 and Hyalp1 are relatively poorly understood. Hyal4 is thought to degrade chondroitin sulfate, but there have been few studies into these two hyaluronidases. We found, however, that Hyal4 has increased expression in bladder cancer. Looking further into this, the Hyal4 transcript is actually a splice protein, so a portion of the Hyal4. This protein, which we've called V1, is missing a part of the normal Hyal4 protein that codes for an anchor that encourages this protein to be secreted by the cell. As a result, V1, this splice protein, stays inside the cell. V1 mRNA was 10-fold elevated in high-grade tumors compared to low-grade tumors and normal bladder tissue. Levels were 10 times higher in muscle-invasive bladder cancer when compared to non-muscle invasive bladder cancer, and levels were 3 to 4 times higher in patients who developed metastasis or who died of their bladder cancer compared to those who survived it. Here you see Kaplan-Meier curves further showing the stratification of V1 levels of patients that develop metastases and those that died of their bladder cancer. In addition, V1 levels predicted response to chemotherapy for metastatic disease. Looking at bladder tumor specimens for patients who had metastases and were treated with gemcitabine and cisplatin, you can see the V1 levels for those that responded well to treatment were much lower than those that responded poorly. Univariate and multivariate analysis showed that V1 levels predicted both treatment response and disease-specific mortality with an efficacy of 85%. V1 transfectants were resistant to gemcitabine, but not cisplatin. Therefore, we've narrowed down where this resistance is. You can see in the top two curves here that these transfectants, when we're exposed to gemcitabine, continue to grow with very little decrease in their growth rate. Whereas if they were exposed to just cisplatin, they were inhibited just as well as the wild type. How could we be impacting gemcitabine metabolism with hyaluronadases? So let's look back at gemcitabine metabolism. Gemcitabine is known as deoxyfluorodeoxycytidine and is brought into the cell through cell transporters CNT and ENT and carried by non-selective drug transporters. Deoxycytidine kinase converts gemcitabine or deoxyfluorodeoxycytidine to deoxyfluorodeoxycytidine monophosphate and deoxyfluorodeoxycytidine triphosphate, which ultimately results in cell apoptosis. When we investigated the impact of V1 on this metabolism, we found that the CNT and ENT levels were not altered, so gemcitabine influx into the cell was normal. The levels of the drug transporters were not altered. However, another enzyme, deoxycytidine deaminase, known as CDA, was at much higher levels. CDA converts gemcitabine, deoxyfluorodeoxycytidine, and deoxyfluorocytidine monophosphate into difluorodeoxyuridine. Difluorodeoxyuridine is an inert chemical. It doesn't do anything to the cell. It effluxes out of the cell, doesn't hurt the cell, the cancer cell keeps growing. So this CDA in V1 positive cells that converts gemcitabine to an inert metabolite seems to be the problem. You can see in this graph that in the colorful pictures here, the two on the bottom right show the increased levels of both CDA and V1. Further studies in the lab showed that V1 upregulates CDA via CD44 JAK2 STAT3 signaling. Fortunately, we have compounds that inhibit CDA and found that using these compounds, we were actually able to reverse the resistance to gemcitabine. The CDA inhibitor tetrahydrouridine resensitized V1 expressing cells to gemcitabine. The bottom picture here, you see tumors injected to mice that were treated with gemcitabine in the V1 positive tumors versus gemcitabine plus tetrahydrouridine, and found that the tumors shrank very nicely. In addition, there was no change in the animal weight or serum or tissue toxicity when these were given in combination. In conclusion, a splice variant, which we call V1 of HIL4, is the first in-class eukaryotic conjoinase that we have found. V1 is upregulated in bladder cancer and drives a more aggressive malignant phenotype. V1 levels in muscle invasive specimens of patients who had developed metastases predicted response to gemcitabine and cisplatin, either given as an adjunct or salvage treatment, as well as disease-specific mortality. CDA seems to play a critical role in this suppression of gemcitabine response, and inhibition of CDA resensitizes V1 expressing cells to gemcitabine. The Lokeshwar lab is a phenomenal lab. Many of you will be familiar with Dr. Lokeshwar's name from her previous work with Dr. Soloway, and I'm very indebted to her including me with this research efforts. Thank you.
Good morning. Thank you for the introduction. I'd first like to thank the meeting organizers for the opportunity to discuss some early work from The Ohio State University that may ultimately deepen our understanding of bladder carcinogenesis and perhaps have implications for the sex bias observed in bladder cancer. Although I've been involved with this work, it has been led by Dr. Deb Sundy, a uro-oncologist in the Department of Urology. He's under the scientific mentorship of Bill Carson of our Department of Surgery and Z. Hiley, a medical oncologist leading our Pelotonia Institute of Immuno-Oncology. He's also had great collaborators shown here from across the campus. Given the importance of immune therapy in bladder cancer, many groups are trying to understand immunomodulation and its relationship to the tumor microenvironment and other components. All of this also trying to relate to therapeutic response. So with that as a backdrop, the objective of this work was to understand the cellular and genomic dynamics of bladder carcinogenesis, particularly in the tumor microenvironment. To accomplish this, Dr. Sundy leveraged the BBN model of bladder cancer, which yields orthotopic bladder tumors induced in wild-type mice after exposure to the tobacco-related carcinogen BBN, as it's put in the drinking water of these animals. He essentially is built on prior studies in the literature now by using single-cell sequencing technology to understand how individual cell populations change during carcinogenesis. So in this study, 12 female mice were exposed to BBN ad lib in drinking water for up to 20 weeks. We monitored bladders radiographically using a micro ultrasound. Here you'll see a control of the bladder, and here at 17 weeks, you see that real thickening of the bladder. At 5, 10, 17 weeks, and also 23 weeks, after the start of BBN exposure, bladders were harvested and processed into a single cell suspension. Histologically, we observed dysplasia at 10 weeks and also carcinoma in situ by 17 weeks after BBN exposure. Bladders were minced, whole bladders were minced with collagenase to create single cell suspensions. Then the sequencing data and the resultant cell clusters were visualized on UMAPs. Now these UMAPs are essentially data visualization tools that cluster similar groups of individual cells. Each dot in the UMAP represents a cell. And these clusters or islands of cells all have different gene expression. Now, from our single cell RNA-seq data, we were able to identify distinct epithelial, stromal, and immune cell clusters based on the genetic expression. So the major clusters we saw, urothelial T cells, stromal cells, and of course, a number of other immune cells. If we focus only on the urothelial cell cluster, we were able to observe expression of druggable targets over time. So there was increased expression of EGFR, FGFR3, and mTOR. We also noticed increased expression of cytokines IL-18 and 33, both which are important in recruiting immune cells to the tumor. But one of the most interesting changes that we saw over time among the immune cells was a big increase in the level and number of T cells. And you can kind of see this T cell cluster here, and over time, having an increased number of cells, increased intensity of cells because of this AR expression. Now, androgen receptor has been implicated in the development and progression of bladder cancer in the past. That's largely happened in studies of animal models, but there's also been some clinical data implicating androgen signaling in bladder cancer incidence. And this comes from secondary analysis of the PLCO trial, where there was a lower risk of bladder cancer incidence in men taking finasteride compared to those who did not. So that suggesting that taking finasteride, which inhibits the conversion of testosterone to dihydrotestosterone, which is a more potent androgen receptor agonist, may have some implication in the development of bladder tumors in these men. Now, Ed Messing and some of his colleagues also implicated androgen receptive signaling in BBN-induced carcinogenesis almost 15 years ago. In their studies, 92% of wild-type males and 42% of wild-type females developed bladder cancer after BVN exposure. But in androgen receptor knockout mice, no tumors developed in either male or female mice. And from our model, in parallel with increased androgen receptor in the T cell cluster, we also observed increased expression of genes related to T cell exhaustion in the progenitor and also terminal T cell clusters. Now, T cell exhaustion leads to attenuated effector function and effector cells as cytotoxic CD8 positive T cells essentially are differentiating into this dysfunctional state of exhaustion. And then as a result of that, they're unable to control tumor growth, and that can result in tumor progression. This is also important because if you have cells where the progenitor and terminal groups are exhausted, then immune therapy is less likely to be effective. So Dr. Sunday is actually now trying to tie these two concepts together, androgen signaling and also T cell exhaustion, in a DoD-funded career development award based on this work and some other work that I didn't have a chance to show you. But that data suggests that androgen signaling may directly contribute to the CD8 T cell exhaustion, some of which we're seeing here in this UMAP on the right. So I find this work to be very exciting. I think from it, we can conclude that the BVN model does have implications for our current bladder cancer research. During bladder carcinogenesis, androgen signaling pathways do increase significantly within T cells. T cell clusters also feature some evidence of gene expression that does relate to T cell exhaustion. And from these findings, really, I think we can say it supports the emerging link that's been in the literature more recently between androgen signaling and T cell exhaustion and bladder cancer pathogenesis. So, from this, we're testing now the hypothesis that androgen receptor regulates key transcriptional activities in effector immune cells in bladder cancer. I'd be happy to answer any questions. Thank you for your attention.
Thank you for the opportunity to present our work. Current therapy for metastatic bladder cancer is effective, but as the slide shows, has room for improvement. This therapy can be improved by either discovering new approaches and new drugs, or to discover new ways to enhance the current therapies that are shown in this slide. Today, I would like to show you what we have done to try and enhance current cisplatin-based chemotherapy. Tumor resistance to therapy is Darwinian. Tumors are heterogeneous, composed of malignant cells harboring different genetic and epigenetic alterations. Treatment selects or creates resistant cell populations or clones. These are either cells that are intrinsically resistant or have acquired resistance to the drug that has been used. Given these findings, we had a hypothesis with three elements. First, that there are specific intrinsic or acquired mechanisms that allow cells to survive cisplatin-based chemotherapy. Second, that we can discover these underlying mechanisms. And third, that we can use this information to target these therapeutically to prevent emergence of resistance and thus enhance response. In order to test this hypothesis, we had to develop the reagents and then characterize these reagents. For this, we developed bladder cancer cells that were resistant to cisplatin, gemcitabine, or both drugs, and profiled these by DNA sequencing, RNA sequencing, or whole cell proteomics, as well as carried out a functional genomic screen to identify targets for therapy. The functional genomic screen to define the mechanisms of chemotherapy resistance is cartooned here. The goal of the screen was to identify the pathway A and pathway B, cartooned here, that keep the cell alive during gemcitabine or cisplatin. Eventually, blocking these pathways will result in cisplatin and gemcitabine working better. This phenomenon is hypothetically cartooned here. Let's say we have a cell that we treat with gemcitabine and cisplatin, but it has a pathway A that keeps it alive. The cell will therefore be alive during this therapy. Similarly, if we only treat the cell with inhibitors to pathway A, the cell will be alive. While if we treat the cell with drugs that target pathway A and as well gemcitabine and cisplatin, the cell will die. This is what is shown here. We took 5 human bladder cancer cell lines that are resistant to gemcitabine and cisplatin and infected them with a lentivirus library that contains CRISPR constructs that target the entire genome. These cells were then grown in either a saline arm or a gemcitabine cisplatin arm, and then we isolated the DNA at various times after infection and sequenced to identify which cells have the various constructs. This screen and sophisticated bioinformatic analysis of the DNA sequencing allowed us to identify a set of core 46 genes that, when inhibited, allowed gemcitabine and cisplatin to work much more effectively at killing the bladder cancer cells. These 46 genes are listed here, and shown in this heat map is how depletion of these genes makes the cells more sensitive to gemcitabine and cisplatin. The next challenge was how to prioritize for further deeper investigation these 46 candidates. We used the criteria that is shown on the left to prioritize these hits based on novelty, druggability, and potency of their synthetic lethal effect in the context of gemcitabine and cisplatin. One such gene emerged, which is NPEPs, and it was a very interesting gene because it was a one of 13 aminopeptidases, and it is localized both to the cytoplasm and to the membrane. Then the reason for these being underlined will become clear in the next few slides. It degrades brain enkephalins and is also relevant in the cell cycle in mouse cells. And you can see in this graph that when it is inhibited, cells become a lot more sensitive to the drugs. We then went on to determine if NPEP's inhibition affected sensitivity to cisplatin or gemcitabine, or both. We used a NPEP's inhibitor called tosatostat to determine this, and found that NPEP's inhibition only increased sensitivity to cisplatin and not gemcitabine. This led us to examine the effect of NPEP's knockdown on tumor growth and animal survival in vivo. We found that NPEP's knockdown delayed tumor growth, and this was further enhanced by gemcitabine cisplatin. NPEP's knockdown and GC led to similar survival outcomes, and these were superior when these two were combined. Finally, NPEP's knockdown was sustained throughout the experiment. Encouraged by these results, we next sought to determine if inhibition of NPEPs could reverse resistance to gemcitabine cisplatin in patients. To do this, we developed 3 patient organoids from tumors that had survived neoadjuvant GemCis in bladder cancer patients. As you can see from the slide, when tosatostat is added, there is far less growth of tumors in the organoid setting than when tosatostat is not added and only cisplatin is used. So how does NPEPs promote cisplatin chemoresistance? Well, not much known in the literature regarding this, and therefore we had to start from scratch. To do this, we hypothesized that NPEPs has to bind to other proteins, and therefore used whole proteomic analysis of NPEPs binding partners to determine these. When we did this, we were stunned to see that the top hits in the proteomics were all part of a same family, a family called LRRC8, which is composed of multiple molecules that make up channels in the cell that are responsible for 50 to 70% of cisplatin and carboplatin import into cancer cells. This was very exciting. We then looked back at our screen and were very excited to see that LRRC8A and D were genes in our screen that, when depleted, made cells more resistant to cisplatin and gemcitabine, which was predicted from the model shown in the previous slide. We then needed to see if NPEPs connected to LRRC8A to regulate cisplatin import. Therefore, we engineered cells with loss of NPEPs, loss of LRRC8A, and loss of both. And this histogram shows the amount of intracellular cisplatin that is in the cells. And you can see that the parental is here in green. The dark green is control scrambled sRNA depletion. And orange is NPEP's depletion, which increases the amount of cisplatin in the cells. But when you deplete LRC8A, it reduces the amount. And when you deplete both NPEPs and LRC8A, what it shows is that you do not increase it further, which is proof that NPEP's effect is via LRC8A. So in summary, we have discovered a new mechanism of cisplatin resistance in cancer. This mechanism is mediated by MPEPs, but is mediated by MPEPs via the VRAC channels LRC8A and 8D, as shown in this cartoon. So the three scenarios where you have drug resistance is if you have fewer channels or channels that are high in number, but MPEPs is present to block their function. Future direction includes understanding the exact molecular basis of the NPEPs and VRAC interactions. We are in the process of building and discovering new and more potent NPEPs inhibitors and considering a clinical trial with tosatostat or better NPEPs inhibitors, depending how fast we can bring those to clinical development. Thank you for your attention, and I am happy to take questions.